Well, welcome back, everyone. I'm really pleased to be joined for a conversation with Bram Verstock. Bram will be well known to many of you, a true rising star in IBD globally. Bram is now consultant gastroenterologist at Leuven University Medical Center and a very prolific author and researcher and thought leader in inflammatory bowel disease. So welcome, Bram. It's a real pleasure to have you with me today.
Thank you, Charlie. Very thanks for the kind invitation.
So, Bram, what I thought we'd do is talk a little bit about precision medicine in inflammatory bowel disease. We've spent this morning thinking about predicting outcomes in IBD, a bit about preventing IBD. We've been thinking about how we can predict IBD onset. We've been thinking about precision medicine as a whole, the role of genetics, and looking at how we can think about positioning therapies by markers, right? So what drug for what patient at what time, and thinking about who needs therapy. Can we predict outcomes A, B, or C for an individual patient? And so we've had this huge hope around precision medicine. Where will it take us? Will it give us the answers to truly personalized therapy for IBD? But perhaps start by telling us a bit about what you think we mean by precision medicine and how that applies to IBD.
Yeah, I think precision medicine, all the different topics you've just mentioned, I think they all cover the very large definition and spectrum. I think it's about diagnosing very early and maybe in a pre-diagnostic phase, I would say. And if you have identified the patients who might be at risk, that you can try to intervene as safely and efficacious as possible so they don't run into the troubles we see, unfortunately, with many of the patients we have. And if at a given stage we need to start therapy, that we then indeed can tailor the therapy and the treatment algorithms as rigorous and as individualized as possible. So to me, it's as you mentioned, it's a whole spectrum of very different phases of the disease, and in all these different phases, precision medicine is, I think, key to improve ultimately the quality of life of patients.
Yeah, I like that. I mean, to me, it's really about how do we individualize care and stop treating everyone with IBD the same? Because we recognize this as clinicians in the clinic, day in, day out. I mean, I spent this afternoon doing the IBD clinic. We've got multiple different patients. Everyone's got their own different set of problems at multiple different levels.
Indeed.
But to me, the precision medicine bit, as I understand it, at its most fundamental level, takes us, I think, down to really a biological level, to a molecular level, where we can start to think and apply tools from the omics era that we now find ourselves in to deploy here. And that's the bit that I find in my clinic day to day I'm not really doing on a regular basis. But perhaps next, let's just have a think about the tools. What are we looking at? What is it, the bits of precision medicine to get us there?
Yeah, I fully agree. So you just mentioned that in our clinics at this stage, we're not using all these tools we should be using because they are not yet there to be clinically implemented, I would say. So we now have to rely on clinical features, and we all know that they can help us to a certain extent. But I think everyone treating patients with IBD agrees that there is not just one Crohn's, there is not just one type of UC. So we have to really dive into disease biology and understand much more what is driving the disease in a given individual, either at a pre-diagnostic phase or at a stage of a flare or whatever time within their disease course they're actually in. Because to me, precision medicine is also about the dynamics. It's not because a specific marker can be very relevant at a given stage in the disease that it will still be relevant at a later stage of the disease. So then to zoom into what are we exactly looking at, I truly believe that as a first step, we have to characterize as deeply as possible from various angles, because it's very difficult to guess what would be the right omic, I would say, what will do the job. And genetics have learned us a lot so far in IBD. They have led to novel therapies. But so far, unfortunately, they haven't really helped us to for instance, tailor specific treatment algorithms, I would say.
Yeah, I would agree with that. I mean, I've been working in the genetics of IBD really since I started working in the field back in 2003. And there was a lot of hope and hype and promise. And, you know, as we've heard from Laura, I think we really have achieved a vast amount. And IBD has been the poster child of complex disease genetics throughout, from the linkage studies to NOD2 discovery through to the early GWAS, the meta-GWAS area, the immunochip era, and then now through into whole exome and whole genome sequencing. You know, and to have over 300 independent susceptibility loci characterized, to understand the biology in more detail, and to see the overlap between gene targets and our therapies that we currently use. You know, just look at IL-23 as a target for a reason. That pathway is stacked full of gene susceptibility targets, JAK-STAT signaling too with our, you know, pan-JAK and JAK1 selective inhibitors. And there are many more exciting aspects that come there. But we're not really seeing that in the clinic yet.
No. And I think there as well, so if you look at all these susceptibility genes, which have helped us a lot in understanding what is IBD, still it doesn't help us, as said before, how to distinguish patient A from patient B. And if we then look at polygenic risk scores, they haven't helped us so far to even diagnose IBD, I would say, because there is so much more. And this is the beauty, but also the complexity of IBD, the interplay between genetics, the immune system, the microbiome, other environmental factors. And that's why it's so key to collaborate in a multidisciplinary approach, both clinically as I would say also translationally, to dive into the omics from various perspectives to really tease out what is important in the biomarker development.
The genetics, let's stick with the genetics for a minute, because it's what we've been doing for the longest. And I think, you know, we understand it really well. Methodologically, we understand it. We increasingly understand the architecture. We have the tools statistically to unpick this. We have the biological tools to start to unpick function. But exactly, as you say, I mean, we did this big study a few years ago, we published in The Lancet, where we did this huge genotype-phenotype study. And we found remarkably little, despite looking in 17,000 patients with Crohn's disease and a similar number with ulcerative colitis, apart from a couple of things that really we already knew. And I wonder, because we know the genetics, but I wonder, do you think we've got the phenotypes ascribed appropriately? Because if we're trying to pair up, for example, genotype and phenotype to come up with some kind of precision medicine marker, we need to have appropriate granularity at both ends.
I fully agree, and that's always a big issue when you're doing large studies with a lot of patients recruited from various time points across the world, that phenotypes are key. And of course, for instance, if you consider the Morial classification and a B1 versus a B2 or a B3, it all depends on how extensively you're looking for it. So we know that a lot, for instance, of the patients with Crohn's disease, they have strictures but are fully asymptomatic, and maybe they are a B2, but they never have been diagnosed with it. And then they are categorized as a B1, and that's where it all starts if you then look for genetic phenotype associations. And I think that the study you were referring to clearly showed that there might be indeed an association with disease location, which is much more stable throughout the disease course, which is also a huge difference with behavior in Crohn's disease.
And it's not particularly helpful either, because we don't have a problem determining where the disease location is. We have the imaging and the endoscopic and biopsy tools to do that. What we really want to understand is behavior, number one, and response to treatment, number two. So where do you think we've got to from that perspective, from both of those angles, Bram?
I think genetics have clearly helped us to some extent. I would say mainly on the safety aspect. We have the TPMT, which is, I think, a very robust and widely implemented marker. The same applies more and more to the haplotype identified in the PANS, where it's clear that if you're considering anti-TNFs, that it might be good to know that haplotype in particular before treatment initiation. Apart from that, in terms of genetics, I think the contribution to precision medicine so far is unfortunately, I would say, disappointing. In other omic fields, there is, of course, a lot of expectations, I would say, on the transcriptomic whole blood marker, initially identified in Cambridge and now being tested in the profile trial across the UK. They just stopped recruitment a couple of weeks ago. So I think all of us are very eagerly awaiting the final results. And if that trial turns out to be truly positive, this will be a landmark trial. And I think the first very robustly studied biomarker in IBD so far.
Yeah, absolutely. I mean, it's really exciting. So a huge credit to Miles Parks and James Lee and Noor and the rest of the team in Cambridge for driving this to completion. The screening phase ended just before Christmas. In fact, I was in clinic recruiting to taking some of those screen pass patients through into the study just now. So it's a really exciting result. So we'll look for the outcome of that. So yeah, I agree. So we've got things like TPMT, we've got the NUT-D15, we've got the PANTS HLA haplotype, which I think we still need to translate to the clinic properly to understand its true utility. Seb Shadji and his team in Hull have been doing some very nice work implementing that into clinical practice already. And then, of course, we've got the transcriptomic markers going through into the profile study. Now, you've been very much taking a slightly different approach to transcriptomics, looking in biopsies and looking to see specifically, I guess, in terms of response to treatment. And that's something that we've had a lot of hope for. So give us a sort of brief update on where that's going, Bram.
I think there are a lot of promising markers there also, again, in the transcriptomic field, including oncostatin M, which was originally identified by the Oxford group. We have the TREM1 marker in tissue and in whole blood, which was initially reported both by the group of Judah Showers and also ours, with a lot of conflicting data and a lot of new data coming out in the next couple of weeks at ECHO and the following conferences. So I think a lot is at this stage is happening also in that era. My concern more and more is that basically, given the complexity of IBD, presumably I would say a single gene marker is not sufficient to capture the heterogeneity and the complexity of the disease. And I think we all ultimately will have to move to panels of markers, as for instance with the profile trial, where it's not solely related or depicted to a single gene or a single protein or whatever. In the same era of the prognostic field, I would say there is also now a new trial to be launched in Scandinavia, led by Jonas Halvarsson, where they looked at proteomics, and where they will take a slightly different approach than the profile trial, but where the aim ultimately will be, again, to prove with a few proteins at diagnosis to prove that you can actually predict up front whether or not a patient will have a mild or a severe phenotype.
So where do you think we're going to see the next real breakthrough to change clinical practice, and how long do you think we're going to have to wait till we can see that?
Well, I think we all hope that the profile trial will turn out to be positive. If that's positive in a year from now, then I think we have a first big breakthrough in the precision medicine field. When it comes to therapeutics, I think a lot of work still has to be done. We have now a few markers, as mentioned before, which have been replicated in smaller cohorts. But the next steps will be to validate this in larger cohorts. And then there is a lot of debate whether or not at a certain moment you should just give it a try as a proof of concept, or really run like dedicated trials as the Cambridge team has done for the profile study.
Yeah, exactly. And then that takes a long time, and you're then potentially delaying the time to clinical utility. And meantime, I think it's worth acknowledging that the therapeutic landscape is moving at a ridiculous pace. I mean, by my reckoning, we have in the next 2 or 3 years 7 or 8 molecules, new molecules, separate molecules, that should enter the IBD space. We also have, biosimilar versions of both ustekinumab and vedolizumab to look forward to within the next 3 or 4 years. So combinations of therapies are going to become possible. So I think we really are going to be in this acute need to really get some better handle on how we position our therapies. How much do you see the sort of precision medicine versus head-to-head study debate playing out? I mean, how much as clinicians are we going to rely on one versus the other?
That's always it's a very interesting discussion, I would say, because obviously we all have been asking for a very long time for these head-to-head trials, and they are still very informative on a population level to give us some idea on positioning of certain therapies. But obviously for that individual patient in front of you, it basically doesn't matter whether drug A or drug B was superior in a population, because to that individual patient, it might just be the opposite as what the superiority trial has shown. So to me, it's not or and or. For me, it's more an and. So I think these two are very complementary. And these head-to-head trials are absolutely important for payers, etc. But I think even if we have these head-to-head trials, and still precision medicine and predictive biomarkers do have their importance. And then I think there is also a very important task and responsibility for industry to basically embed that trial, that biomarker development and design already into their early drug development programs. And you now see more and more companies, luckily, sampling very intensively during their phase 2 and phase 3 programs. So there is a lot of data out there which has not yet been publicly available, but which could help us a lot to foster basically that era of predictive precision medicine.
Very, very nice, Bram. I like that a lot. To me, that is a call to arms for all of us, for those of us as clinicians, with our academic partners, but also to our industry collaborators with whom we really can't do this. It's not just to encourage them to collect the tissue to do these experiments, but also then to have the courage to go on and do the analysis and report the findings, be they positive or negative. So, I think I could talk to you for hours about this, Bram, and we could cover lots more ground, but I think that's about the time allotted for now. So that leads me to say thank you so much for joining me for this conversation. Pleasure. Just now. I think that gives people just a snippet of just how fascinating this area is. It's really exciting. The next few years in IBD, from a clinical, therapeutic, precision medicine aspect, is going to, I hope, truly revolutionize the lives for patients. And for us as clinicians, we're going to see the impact of that day in, day out back in our clinics. So, Bram, thanks very much for joining me today. And I look forward to catching up in real life very soon.
All right. Thank you, Charlie. Bye, everyone.